Hello, everyone. It is my great honor to introduce our distinguished keynote speaker, Dr. Farah Sheikh from UC San Diego. She is currently a professor of medicine. So Farah grew up in Quebec and Northern Ontario. Excuse my pronunciation. And then Farah went to McMaster University to get her bachelor degree in biology. And then she moved to Manitoba, did her PhD in cardiovascular physiology in the University of Manitoba in Winnipeg. And she worked on the FGF2, fibroblast growth factor 2 in cardiomyocyte survival signaling, a very pioneering work. Then Farah went to San Diego to get her postdoc training with two truly pioneering scholars, Kenneth Chen and Zhu Chen. So Farah got her own lab back in 2009, and she was promoted to associate professor 2014 and to full professor 2020. She has done truly amazing, innovative work in her own lab. So she developed human relevant mouse models, especially on the cardiomyopathy, arrhythmogenic cardiomyopathy, and she has identified key targets for therapies. And she has also developed new approaches for gene therapies for the arrhythmogenic cardiomyopathy, which is now in the clinical trial. And she is also the chair of the BCVS council meeting of the AHA. What else should I? Sorry, I don't have the note. Okay. So Farah, if I forget something that you wish to mention, you can do it by yourself. Thank you. Okay. Thank you.
Thank you so much, Wenben, for that lovely introduction. I feel like I'm coming home. I don't really come to Canada that often to give talks, so it's really nice to come back to familiar, friendly atmosphere. So I won't go more into that, but so I also want to thank Katie for this wonderful invitation, and you know, especially the special occasion of the 50th anniversary of the Research Institute. It's really a privilege to be here. And to be honest, I've been so impressed by all the faculty and the trainees that I've met, like so bright, so sharp, so I mean, it's a perfect environment to do translational science. So I really congratulate you for, you know, honing in this environment, because I don't see this quite often, even at my own institution. So I'm going to put that in there. So today I'm going to share with you my own journey of into translational sciences and how our work has really helped on alleviating some of the burden, at least a little bit of the growing burden of cardiovascular disease. So here we go. So we know that cardiovascular disease, I don't think I have to say anything to this audience, continues to claim millions of lives each year. And although current treatments such as medications, surgeries, devices, lifestyle guidelines have really saved countless lives, we're still a little bit short in terms of the challenges that remain, where we don't sort of tap into the underlying cardiac biology that may predispose us even further to disease. And a lot of it is on symptomatic management. So I work in the inherited cardiomyopathy field, and genetics matter there. And I think it also matters in broad diseases as well. And I think this is sort of the landscape for us. You know, we know the molecular needle in the haystack. Now, how do we change that into a therapy is a question. But now we also have, you know, a potential roadmap to try to tailor therapies for not just symptom management, but going after the root cause. So I don't know if people have seen this sort of diagram. Once you discover a molecular needle in this haystack, how do you go from research into practical outcomes? There's this big valley of death where 90% of innovative technologies, ideas, sort of unfortunately don't make it through this. And so I think this is the big challenge. I think you guys are in a perfect environment to try to solve this because you are working symbiotically with clinical and research together. My own journey really through this valley began when I was really deeply immersed in sort of understanding the genes that are linked to this syndrome or this inherited cardiomyopathy that is causes sudden death in young populations called arrhythmogenic right ventricular cardiomyopathy. And unfortunately, a personal connection really brought this issue closer to home because someone in my life got diagnosed with this very disease as I was studying it. So sometimes fate leads you in a certain direction. And so it was really a stark reminder that it's not just important to understanding the biology, but also pushing the research towards a therapeutic solution. And if you don't do it, nobody else will. And so I think that is the key urgency that I felt because I had this person in my life and I had to potentially tools to maybe try to understand it better. And so this is really what motivates my research to this day, and hopefully it motivates you. I know motivation is a big factor in staying in research, and because there's a lot of ups and downs, but I hope you take this as inspiration to sort of find your deep motivation in driving what you believe is important for research and going through those gaps. So when we think about this disease a little bit more, you know, based on the name arrhythmogenic right ventricular cardiomyopathy, it's been traditionally thought to be a right heart disease, but recent studies suggest that there's also complicated forms where you affect the left side, both sides. And so there's a complexity there to this disease. The other thing is from the name, from the arrhythmogenic part, there's clearly electrical deficits that drive the disease. And you can see this in a rhythm strip. This is just a premature ventricular contraction, but these are made worse with exercise and it can lead to sudden death in young people. And when you look at biopsies, unfortunately, or autopsies from these patients, you can see there's a lot of structural destruction. So very little viable red muscle, a lot of this blue fibrosis that's come in. And what's unique to this disease that there is fatty deposition. And I've heard a little bit about, you know, atherosclerosis, fatty deposition, maybe there's some connection there, but basically this is something unique to this cardiomyopathy. And although it's really inherited, unfortunately it's adult onset. So there's this phase where it's concealed, where you don't really know you may have it until you have someone in your life affected and get screened or have symptoms that, you know, lead to some of the early electrical arrhythmia issues that one has. So this is unfortunately a survival curve. It's kind of old, but it's kind of important to note that, you know, people with ICDs were in this survival curve. And here what they've shown is they've taken a small group of patients and they've shown that 40% of patients die within 10 to 11 years after diagnosis. We've extended that because we can diagnose things a little bit earlier, but this is really, unfortunately, the course of events. And all of the interventions are to the arrhythmia burden. So nothing to the cardiomyopathy that continues to progress with this disease. Of course, transplant is an option, but there's really a high cost and morbidity to this disease. And unfortunately, the first prescription given is exercise restriction, which slows the progression of the disease. And so unfortunately, a lot of these patients are athletic. So that is really an impact on the quality of life of these patients. What we do know, so some positive news, is that it's thought to be a disease of the desmosome. And I'll just go through this. When you look at the heart muscle and you zoom in, there's an area where the two muscle cells connect, and there's really a trifecta of junctions that encompass this. And desmosomes are these very electron dense mechanical anchors that prevent your heart muscle cell from ripping apart. And when you zoom in and look at what are the proteins that anchor this, there's really 5 central proteins, and 40 to 50% of cases are linked to mutations in each one of these genes. The other thing that's interesting about this is that in the end, although you have an issue with the desmosome, it continues to wreak havoc on all of these other junctional compartments. And so it often you have a domino effect of the whole junction being destroyed. So it's been long thought in the field that no one gene can fix it all, because you have to bring in this gene, that gene, now this compartment's disintegrating, that there's really hard to kind of get at in terms of therapeutics. So this was one of the fundamental challenges to the field, is that we didn't really know how to bring this macromolecular complex back together again, what therapeutics would modulate this. So when we went into the field, we looked at, well, what is the majority of the population? What mutations do they have? And they have mutations in a gene called plakophilin 2. It's the most frequent mutation. You can see from this table, 70% of patients here. And as we started digging in more and more into this disease, we found that, yeah, of course you have the genetic mistake, but it's the messaging that's gone awry, because RNA levels of these protein of PKP2 is altered. They've done transcriptomic, and actually the number one PKP2 mutation is an RNA splice site mutation, which clearly affects the message. But when you affect messaging, it's very hard to know how to intervene with therapeutics, because you can either get a larger protein, a smaller protein, no protein. And so how do you deal with therapeutics in terms of this population? And on top of it, there were no models to better understand this. And so we just simply hypothesized that the splicing and RNA messaging is a problem, and that it's going to be sufficient to trigger the disease. And we have to boost RNA levels in order to therapeutically intervene with this disease. And so in order to test this, we actually made that a mouse model, a genetic mouse knock-in model with this number one top splice site mutation. And so these are all some of the papers that show that it's found in multiple populations across Europe and US. And you can see here that if you look at the survival curve, if you have one copy which is deficient, you can see that, you do have survival deficits over time, but this, two hit, as they call it, is where you get significant mortality. And oftentimes we call ARVC as a two hit because you either have to have exercise and a mutation, or two mutations that drives severity of the disease. So this is just some histology on the hearts, and this floppy right ventricle, you can see tons of scarring, fibrosis, and of course this fatty deposition, which we've stained with perilipin and lipid oil red O, I should say, that sort of is a hallmark of this disease. And it's always found in this particular region called the subepicardium that we see. And so this mutation, it was a global mouse with this mutation, but it only had the heart issues, and which was surprising to us in a certain way. But we wanted to understand better what this mutation did. And so the first thing we did is look at the message, the PKP2 RNA levels. And you can see in these PKP2 homozygous mice, you get abnormal processing. You actually get what's called intron retention. So you actually get a bigger sized product. And this product is at very low levels. And so the idea was when we looked at the hearts of these mice at the protein level, again, we see this larger size product and this destruction of the desmosome. So all of these proteins are downregulated, including connexin 43, which is the electrical communicator, which is the one that basically drives a lot of the arrhythmias. So the question was, is it protein quality or quantity that drives the disease? Because this could be a poison gene or protein that wreaks havoc on the cell. And so this is kind of what was the first inclination for us to sort of patent these findings when we did this particular experiment. So these are cells in a dish. And here you have the PKP2 homozygous cardiomyocytes. You can see again this low level of this higher migrating protein, and then it wreaks havoc on this. So there's kind of a blank space because it reduces all of those desmosomal proteins. And what we did is we boosted the level of the wild type protein and also that mutant protein. And what we see is that when we boost expression of both of these, we can actually resurrect the whole complex. And so this was the surprising finding, is that this mutant protein is not really toxic. And we think that's why maybe patients survive, because this mutant form still has functionality. And the idea is it's all about the quantity. We just don't have enough of it there. And third, could PKP2 be the one gene that fixes it all, given that it sort of scaffolds and glues together the cell because all of these proteins get re-expressed? And so this was the scientific premise behind the PKP2 gene therapy that we then went on to test in our animal model. So I'll just share with you some of the initial data. So of course, when we gave it early at day 2, you can see that this is a survival curve going 6 months out, and that 100% of these mice survive. So there's absolutely no death in these mice when you give it preventatively. And you can see in these mice, that get left, unfortunately, you only have the treated and the wild type, that they have robust levels of all of these desmosomal proteins. So they're able to retain all of this, and it was associated with survival. But I think the critical piece is when you get patients, they're probably going to have some level of disease. So how do you whether it would work in late disease was a question mark for us, but we definitely wanted to test that. And so these are what these hearts look like at 4 weeks of age. And we treated this mice, again, a one-time injection with our gene therapy. And we were scared, so we just looked 2 weeks later, and we could see already that when we boost the level of plakophilin-2, now all of these proteins get re-expressed even 2 weeks later. And we did some MRI imaging even 2 weeks later, and now you can see the groups start to separate in the sense that ejection fraction is much better in those treated with the gene therapy versus ones that are not. And so we actually let these mice go for 20 weeks, and you can see that 100% of these mice survived, even if we gave it at this late disease time point, which really fortified to us that this was a potential therapeutic that could work even in a disease setting. And so just to summarize quickly, what I've shown is that these RNA splice site mutation drive effects on PKP2 protein quality and quantity. And we think that mutant form is very important for people to survive. And so I told this to a patient advocate, and they're like, oh, I didn't realize that this actually made me survive, so there's some positive to having the mutation and surviving, but there's clearly a question mark when it comes to thriving, because over time you get continual destruction of the desmosome, which wreaks havoc on the cell-cell junction, and then you can get all of these hallmark deficits, including premature death in these mice. And we believe that this AAV based PKP2 gene therapy highlights that there's some sort of gluing action or scaffolding function to this protein that can prevent and rescue cardiac disease. So how do you take this into research, into practical outcomes? So I patented these findings. You have an amazing tech transfer office here. I went to mine and I provisionally patented these discoveries. And I realized after a year of provisionally patenting them that I wanted to start a company. And so this is ARVC Therapeutics, very intuitive to the disease. And but I realized I needed the right partners. And so this is where I ended up merging my company with another company called GenStem, which was led by Stephanie Cherokee and Eric Adler. They had already taken their drugs to phase 1, 2, and I wanted to as well. And they had started a new company on new assets. And so I joined them, and we ended up merging together to form Stelios Therapeutics in May of 2020. So we, during COVID, were pitching to like people, and that was a whole process. I can go through that, but we were very happy in 2021 to get acquired by a genetic medicine company called Lexeo Therapeutics, where they acquired 3 of our assets, 2 of them being for ARVC and 1 of them being for hypertrophic cardiomyopathy. So there was a critical question, because that company, you know, we had both AAV gene therapy assets, but we also had cell-based therapy assets, which were all related to rare disease. And so we had to make a decision where we ended up splitting the company such that we could move forward with the AAV gene therapy assets with Lexeo. But then we made a new company called Papillon Therapeutics on the cell-based therapeutics. So I work, you know, it's a path that sometimes you have to think about. Nothing is smooth, but you take the path that would get your product the furthest. And I think, you know, we're both happy where we're at now with our with this decision, but at the time it was very nerve-wracking. So we ended up going forward with Lexeo to do proof of concept IND enabling studies using LX20, what they call now LX 2020, and it's using their particular capsid called AAVRH10. I found that this was much more efficacious in the right heart, which is where I wanted the therapeutic to go, and you could go at lower doses. And of course, we used our PKP2 HOM mice to drive that the studies. And of course, we injected it. We basically waited 12 weeks. So again, these are very diseased mice. We gave it for 12 weeks, and we looked at all of the markers of the disease. So first off, I'll just kind of quickly share that, of course, the survival curve, you can see the wild type mice, the mid dose and high dose. They're all doing, 80 to 100% better than any of the vehicle or the low dose. So there was a huge effect on improving survival. There was also a huge effect on arrhythmia. So you can see in the mid and high dose, more mice are arrhythmia free. So this white bar. And even if there was one mouse that had one arrhythmia, we put it in the arrhythmia group. So these mice had very little arrhythmias. And I think I like showing this because it's intuitive. You can see the hearts moving. You can tell these, of course, are vehicle treated mice. These are untreated mice at the same age. And you can see how poorly they're doing, the ballooning of the heart. And as we gave higher and higher doses of LX 2020, the hearts look virtually like the wild types. And this was also coupled with the histology. So you can see here, these hearts are falling apart. And as we gave higher doses of LX 2020, some mice looked like they had never had any injury or disease at all, which was completely surprising to us. So we're trying to follow up as to what's going on. But we know that basically boosting the level of plakophilin-2 scaffolds the entire complex. And you can see in the vehicle treated, there's nothing there. It's pretty much disintegrating. So these data really fortified that it's a promising gene therapy candidate for treating patients with ARVC. It restores not only plakophilin-2, but kind of resurrects the whole complex. And through dose-dependent studies, we're able to show its effects on ARVC deficits. And so all of this was done in my lab, but we coupled with the company, and they did all the toxicity studies in non-human primates to show that this drug was well tolerated and safe and distributes within the heart. So we put this together. submitted it to FDA, and in August 1st, 2023, we were cleared by FDA to move into phase 1/2 clinical trials. And I think one of the big hurdles is how you need a lot of money to run clinical trials. And so we were fortunate to actually be a part of the IPO filing. So they went public and they were able to secure the funding. So this is in the middle of Times Square where we rang the NASDAQ bell. So this, I think, was critical to move forward into clinical trials because you do need a lot of money to do this. And so since then, Lexeo has reported that a year later, they had dosed their first group of patients at the low dose. In March 2025, they had dosed the patients with the higher dose. And just in January, they've reported that across the 10 patients that the LX 2020 is safe and well tolerated, and that we do biopsies before and after the gene therapy. So we're able to show transduction, transcription, and also protein expression at a dose-dependent manner in these patients. And also there is some early signs of efficacy that the arrhythmia burden is essentially improved and or nullified in these patients, in the majority of these patients. And so, this, I feel, renders some hope for this particular population, given that there was no treatments for this patient population. And I think one of the things that I am on a blog for ARVC on a Facebook group, but I was hearing a lot, well, what about me? What about my mutation? What can I get as a therapy? And so we know that not all ARVC patients have mutations that are known, simple, or amenable to gene therapy. In fact, one of the worst populations is with the gene desmoplakin. They have a lot of inflammatory disease. They also have the higher mortality burden. And so I'm going to just share with you that we've worked also in this space and shown that gap junctions and connexin 43 may be a mutation agnostic therapy for them. And so one thing that I'll also mention, why connexin 43? Well, it's thought to be a molecular hallmark of this disease. Most patients have loss of connexin 43. And so this is one of the studies that we showed that when we dose dependently knock down desmoplakin in a dish, that one of the first things that's affected is connexin 43, even before any of the other structural proteins. And this results in conduction blocks in a dish. So there's very early effects of connexin 43. But I think over time, people have done a lot of biopsies on these patients and shown that connexin 43 often tracks with the severity of disease, and that this reduction is also a hallmark of late stage. So it's almost the most downregulated. But no one really knew what it did at this stage, whether it was even important. And so we hypothesized that maybe connexin 43 has roles beyond the electrical sort of canonical functions, and that it may have an effect on the structural alterations that lead to myocardial failure and premature death. And so, of course, you get downregulation of connexin 43. So what we did is we simply restored it in the face of desmosomal deficits to see what it could do. And so this is just some data on this particular in vivo treatment protocol. So we used a desmoplakin deficient model. Again, very, very severe, almost like we mimicked a pediatric case that was very severe for desmoplakin loss. And we simply restored connexin 43 and looked at molecular, functional, and survival analysis in these mice. And you can see without a major protein how putting connexin 43 completely prolonged the life of these mice by twofold. And so we were extremely surprised by that. And we looked to see what was going on in the heart tissue. And again, no desmoplakin, absolutely none. But when we give back connexin 43, we can completely boost the level of all of these proteins and the expression. So again, this sort of goes towards the idea that there's some plasticity with these cell junction genes that we don't really recognize that may be sort of reprogramming the cell in a specific way. When we looked early on in these mice, we were able to show that these hearts have reduced dilatation, improved function in this severe mouse model, and they also have reduction in arrhythmias and alleviates all of the cardiac arrhythmias. So again, this is looking early on in the trajectory. And so we wanted to understand, this is a pretty severe form of ARVC. Can connexin 43 restore or alter these deficits in other forms of ARVC, such as other desmosomal mutations and autosomal dominant? So we did it using our PKP2 HOM model to see, well, could it rescue that model? And also we actually collected, in collaboration with UCSF, skin biopsies from patients with various disease heterogeneity and made iPS-derived cardiomyocytes to look in a dish whether it could salvage that. And so here are the results from this plakophilin 2 mouse model. So you can see these mice, 100% of them survive, unlike 80% death in this model at this stage. Again, this is the severe form using a different mouse model. And you can see that desmoplakin, you know, if it's there, its expression can also be boosted. So this is potentially a way of regulating desmoplakin expression. And again, here, the black bars are tracking with the blue bars, which are the treated and the wild types. And you can see that there's reduced dilatation and improved function in these hearts. We also looked at iPS cells with various mutations in desmogliin and plakophilin 2. So these are, again, and what we found is that in the untreated, the connexin levels seem to track with severity, as was shown in a lot of mouse models and patient tissues. And when we look at some of these electrophysiological deficits in a dish with coupled cells, you can see a lot of baseline issues in the plakophilin 2 model. And when we give isoproterenol, this becomes worse. But this desmogliin patient only had exercise-induced arrhythmias, and we could see the deficits only with isoproterenol. So suggesting that it mimicked sort of this disease in the donor heart. And of course, when we gave connexin 43 back, it completely normalized the signatures in these cells, whether at baseline or in a isoproterenol setting. So what's going on? How is this potentially affecting the sort of health of the cell? We actually looked at both iPS-derived cardiomyocytes as well as mouse models. And of course, we're transcriptionally boosting connexin 43 to either overexpress it or normalize it. But what we were surprised by is that we're also boosting the level of all these other mechanical genes. And so we were trying to understand, how is it doing this? And there was this paper in frog oocytes that suggested that it could be a cofactor, a transcriptional regulator of mechanical genes such as encadirin. And so I won't go into the too much details, but we cut up the promoter. We put connexin 43 along with polymerase 2 and found that there were areas of the promoter that are closest to the start site that would boost the expression of these genes. So using CHIP PCR, we were able to show that it could act as a cofactor to boost the level and synthesis of these mechanical genes to sort of resurrect the whole cell back together again. So I think in summary of this work, we're able to show through various mouse models, iPS-derived cardiomyocytes, that when you have deficient or destroyed desmosomes, connexin 43 is one of the first hallmarks. And if you restore this at early or late stages, you're able to boost the synthesis of these genes, which then has a huge effect on the electrical coupling and the function of the heart, as well as the survival of the mice. And so we believe that it may not just have electrical functions, but it also may have mechanical functions that were previously unknown. So just to summarize, we've tried our best to fill some of the critical gaps. Of course, we've understood that PKP2 gene therapy could be a great way to restore function and survival and improve health of patients with PKP2 populations and mutations. And we believe that Connexin 43 has an effect potentially in a mutation agnostic way that could be used for the broader population in bringing back the cell back together again. So just some of my final thoughts on some of the biggest lessons that I learned. First of all, step out of your comfort zone. I think that is one of the big things that I learned. I mean, of course, I had someone in my life that was affected by ARVC, and so it pushed me over that. But I think that's often the first hurdle mentally that you have to get over, that I want to do this. And so that's one of the biggest lessons. Also, embrace mentorship. I think, when we think of translation, it's often a marathon where you're passing the baton to someone, they're passing it on to the next person. And so there's a lot of mentorship around that. And I feel you don't really learn this in the lab per se, unless you've gone through it. And so learning from other people that have gone through the process is helpful, but also your tech transfer office, leveraging all of your existing resources. I know Canada has a lot in that space to help people with startup companies move things forward, give advice. I mean, at our institution, we have entrepreneurs and residents who are people that they've retained from the outside to help entrepreneurs make those decisions. And sometimes, they're often the people that push you over the edge. The other thing is, if I hadn't merged my company, I don't know where I would be with my work, because I would rather take an equity cut and be with them and learn from them to make sure that I moved my, my product forward. Because you have to sort of recognize that you can't do it all. I think now everyone wants to do it all. Everyone wants to have everything. But I think there's a lot of give and take in that process. And you have to be willing to recognize that you can't do it all and that you need mentorship through all of that. So I'll end by just thanking my lab, who have been instrumental to a lot of the work. I've highlighted Jing Zhang and Matthew Ellis and Fabian, who were instrumental to the Connexin work, and Hank, who was my lone graduate student working on the Plakophilin 2 story, who's now in industry. Surprise, surprise. But, was a really, it's a collaborative effort and it's a team effort. And I feel like, if you have that and that support, it's what's needed to move you through. And of course, my collaborators. I can't be more thankful to UCSF, my clinical collaborators there, who gave a lot of the biopsies and also support to the work. And I think, I was also very skeptical of industry, but I think there is a way, and there's now an ecosystem where everyone's working hand in hand, academia, investors, industry. And so there's a much more collegial back and forth between that system. So I'm grateful to my partners at Lexio, who have helped me move some of the therapies forward. So I think with that, I'll stop and I'll take any questions.
Wow, it is truly amazing work. Congratulations. So we do research because we want to help the patients. That's the only purpose that we come to the lab every day. And yeah, anyway, I will not say that much. I'm always interested in great research. I do have 3 questions, but because I'm the host, I will let the audience to ask questions first. Sorry.
Thank you, Farah. That was very inspiring. We talked yesterday, you and I, about some of the imaging hallmarks of this disease. It has obviously very many phenotypes, but One of them, which you mentioned in your presentation, is it tends to affect more the epicardium or midwall. So I have a couple of questions related to that. Are these junction proteins more expressed in those areas of the heart, or do you think it's more like the shear forces when the people exercise, it just pulls more on these junction proteins because of where they're located in the myocardium? And if so, do you think there would be a role for beta blockers, to help, in addition to your therapies, like targeting, reducing shear stress?
Right. I mean, that's the holy grail question. Why is it in the one location? I think, to be honest, people have done studies where they looked at the amount of it at those locations. They don't seem to change per se, but it doesn't absolve functionality changes that could happen at those locations when you have mutations. There's clearly different workload distributions across the heart that we, bioengineers, I've worked a lot with bioengineers on this front, and that could play a role into it. I also think, it's let's put it this way, that because it's so out of the, like, at the very surface of the heart, there are other, like, fat and inflammation and all of those things that could play a role in driving some of the subclinical etiology that is at the surface of the heart per se. And beta blockers, they are using that on these patients. So but I agree. It's got to be, even in the trial, I mean, everyone's on their regular medication, but then they're taking this as a,
Thank you.
Thank you very much. It's an amazing talk and a very inspiring and very, very nice data. My question is, in the PKP2 knockouts and in conditions where you overexpressed or knocked down connexin 43, what do you think that signal that the cardiomyocyte get so that it change all this transcriptional profile of other proteins? Or do you think this is something related to the milieu, whether the desmosomal proteins are not stable anymore?
Yeah, so there's a lot of thought around that. I would say that, the transcriptional thing was a little surprising to us, to be honest, because I thought it was more about stabilizing what's there and preventing degradation per se. We actually uncovered a few degradation machinery that was specific to the cell-cell junction. But I think, connexin has nuclear localization signal. And so there is some, and we did find it near the nucleus. And I think that is something that is more and more being looked at, the trafficking role of connexin 43. So I think we always think of some of these proteins like at one location, but I really think it's much more dynamic than that. And that having this toggle between the nucleus, I mean, I think the cells are kind of being reprogrammed in a way to fortify some of these mechanical junction genes. So I think we're still trying to uncover, could it be opening areas of chromatin that is more permissive to some of these expression of these genes? We're looking more deeply into that. We don't know the full answer. Yeah.
Again, I'm absolutely not an expert. First, it was an amazing presentation. But what's happening on the left side? There is a compensatory mechanism, or why it's only on the right side?
Yeah, so that's an interesting question as well. That's also the conundrum of the disease, like why right heart and is the left? I mean, some genes are more permissive to left-sided disease, such as desmoplakin, whereas some genes like plakophilin 2 are more right-hearted. And I don't know the answer to that. We've tried to look for microRNAs that may regulate genes specifically in the right versus left. I think I've heard also potentially the immune response may be a little bit different between left and right. So we're looking at all these combinations. I think it's merging between cell types. I mean, we're getting more and more data in our particular models that it's interaction with other cell types that may drive potentially some specificity towards one chamber or another. But I think, we're very cardiomyocyte centric, so we haven't looked at that. But this is kind of the next phase of us trying to understand some of those processes.
Thanks, Farah. Absolutely inspiring. Thank you so much. And a fabulous journey that you've taken us on. My question, I had similar questions about the LV, but
Yeah.
this is more now pragmatic question about how you took your idea and convinced investors to invest in this to help you cross the valley of death.
Well, I have to say timing.
And what advice would you have for
Yes.
the entrepreneurs and the scientists who are having their ideas, their startup companies, what they need to do?
So I think, to be honest, we were lucky in the sense that the timing was right. When we were pitching, like, gene therapy was huge for cardiac. People were looking for cardiac assets. And the company that we partnered with, they had another program. They had brain and heart related programs, and they didn't really have a lot of expertise in the heart, although they were going in the heart, and they had just come out of stealth mode. And we just kind of came into their path because we failed out of one deal and someone actually introduced us to somebody else. So it's often your failure that leads to your success, because, we pitched to like we got 5 offers, 4 of them fell through. And because we had such a rapport with some of the other companies where things fell through, they were highlighting us to other people. And so I think that's why I think it's very important to have a good network and always keep, good faith between whenever something doesn't happen the way, because probably meant to happen in another way. And so I feel like people lose hope when they're like, oh, it's not going through. It's not. But like, I feel like we had to do an autopsy after every failed deal, let's put it that way. And we always learned something new about how to do a better job next time or what to hone in. So I think that is, I don't know, the art of the deal. I guess there's a book on that somewhere, but I think, as entrepreneurs, you're going to go through a lot of experiences and highs and lows. And you have to be able to be resilient through that, come back with fresh eyes, fresh thoughts, and just network with others that are in your same space and keep those networks alive. Let's put it that way. I feel like that's part of the way that we ended up.
Quick one. I understand this much less than probably most in the room, but for ventricular arrhythmias, you need both, a substrate or the tissue to be able to support the arrhythmia, as well as a trigger. And you've described how this really changes the substrate or the underlying disease. But do you have any understanding of whether this is impacting the rate of triggering? Is this also affecting anything on that Purkinje system or anything to that degree that would change the rate at which some of those triggers may occur?
Yeah, so we've worked a little in the cardiac conduction space. Connexins are also found in these conduction cells, right? So we haven't done the subclinical looking into seeing how that affects those connections, which might be important towards not only the substrate, but the triggers that may drive this. Purkinje in particular are very open to connexin 43 biology and function. So we think we're affecting that as well. It wouldn't exclude that per se.
Thank you.
Very informative as well as inspirational presentation. I have 3 separate questions. Number one is that in this very rich research conference, there are 83 posters out there. And I spotted one of them, which may be quite related to your finding,
Okay, let's
Um,
hear.
it's 44. It's called the high incidence of pacing-induced myocardiopathy. Now, that the result was that a higher pacing of the right ventricle increased the myocardiopathy. I wonder if this is anything connected with you, like for example, that higher pacing results in a destruction or a degradation of your testosterone.
Yes, I would say yes. So we've worked with some bioengineers on looking at pacing and disease burden. Definitely exercise would be the typical thing where you're increasing heart rate and it may trickle. I think that's why you get less disease when you stop exercising, which could, affect the pacing and the workload on the heart per se. So I haven't looked deeply, but I think that is something that, the biomechanical stress, like that's one of the things that we need to better understand across the heart, not just an aggregate force, but like that there are spatial layers that have specialized workload that may define why one would get disease, especially with desmosomes that are very biomechanically stress sensitive.
Now, this will be highly significant because that pacing of the right ventricle is very frequently happening, particularly in the regular pacing of the heart, normal pacemaker, not ICD. And the changing of the mode to MVP versus, the DDD.
Right.
That has a high effect, and this will be very significant to find out.
Right.
Now, the second part of the question is coming back to your entrepreneur. I keep hearing that you're saying you're using a drug, whereas it's a genetic therapeutic.
Yes.
Now, here it could be a public image problem. Is the genetic therapeutic really has a similar effect as drugs? Because drugs have side effects.
Right.
All right. I don't know. Okay, I'm just asking. This raises my question. Does the genetic therapeutic has side effect like drugs or no?
It does.
Okay.
There could be an immune response related to using gene therapy. That's why you kind of have to use it at a certain dose because it may trigger toxicity to the liver. And also if you use it high enough, unfortunately, there's been some deaths with gene therapy which relate to complement activation and things like
Yeah,
that.
that's too bad. Otherwise, I would think that you really have.
Well,
Okay.
nothing is without side effects.
Okay. Well, devices
Okay, maybe
devices may not. This is the thing. Now make it unique, right? Now, the third one is that since you're entrepreneur, you're encouraging that. And normally, again, with the corporations, people again has the image that they're going for profits. Now, could you people, like starting from academic and the idea then become commercialized, to have a special influence beginning that, if I'm successful, we're going to have a special branch and label your particular things with that's a philanthropical benefit. That might be a good thing to go.
Yeah, I mean, absolutely. I've now supported other startups because of my success. So I think that's why what goes around comes around. Like you feel like you need to push other entrepreneurs as well. So I think that's the positive that comes out of it.
Well, make it public so people know.
Oh, okay. Okay. Thank you.
Okay. My first question is the wall stress experienced by the mouse heart, is it similar or the same as in the human heart?
Wow. Well, I believe so. I think that there is precedent for showing that the workload differential is similarly experienced. Now, I don't know about the exact numbers, but definitely there is this workload stress across the heart in the human and mouse.
Okay, thank you. So next question, in the animals that already have this heart damage and
Yeah.
also in patients, when we use the viral vector to
Yes.
express the PKP2,
Right.
doesn't, I mean, prevent further damage to the heart, or it can also repair the already happened damage to the heart?
Yeah, so I think we're looking into that. So I don't know when we looked in the, there's some pliability to the mouse heart. I'm hoping we see that with the human heart, but I'm I guess cautiously optimistic to see that reversal. I think that's where we're really trying to understand how is it really reprogramming? Is it changing the course of what the fibroblasts are feeling? The interesting thing is that connexins and PKP2, like desmosomes, are also found between myocytes and fibroblasts. They're not just found between myocytes and myocytes. And so there could be some coupling deficits that we are also repairing in that space, but I think to be determined.
Okay, thank you. Last is actually a comment. I think after this viral expression of PKP2
Yeah.
protein, for example, only part of this protein is needed for
Yes.
this desmosome repair.
Yeah.
I think there will be a free pool in the cytosol that can be used, or just like troponins,
Yeah.
to measure the serum level of the PKP2 to see
Oh, okay. Yeah.
as an index of heart cardiac tissue injury. I mean,
Yes.
the
Absolutely.
progression in the animal models,
Yes.
if the damage is reduced, slowed down. Yeah, I mean, just a comment.
Yeah, no, that's a great point.
Yeah, thank you.